Hello everyone, and welcome to this short video from the Foundations in Neuroscience series. I'm your host, Professor Phil Newton, and in this video we're going to talk about action potentials. These are the basic electrical currency of the nervous system. It's absolutely fundamental to understanding how the nervous system works, is understanding the basis for and the propagation and generation of these electrical signals that we call action potentials. What I hope you're able to do then by the end of this short video is to be able to describe the firing of an action potential, the very basics of it. In order to understand what an action potential is, we need to go back over briefly the biology of the main electrical cell type of the nervous system, and that's the neuron. The action potential travels down the neuron in a very specific direction, and we need to talk through the main components of the biology of a neuron in order to be able to understand that. Here then again is our neuron with its action potential traveling, starting at the dendrites where the action potential comes into the neuron, then processed in the soma or cell body. The electrical part is generated at the axon hillock. It travels down the axon and leaves the neuron at the axon terminals. These yellow blobs are the myelinating cells that produce myelin, the electrical insulation for the axon. And we're going to talk about these and all of the other components of the neuron as we make our way along the generation and propagation of an action potential. The starting basis for an action potential, then the means by which a neuron is poised and ready to fire or receive and then fire its own action potential, is what we call the resting membrane potential. There is basically an electrical and a chemical force acting across the neuronal cell membrane. The electrical force results in the outside of a neuron being positively charged relative to the inside, and the chemical force means that this electrical charge is generated by there being chemical concentration gradients of various different ions across the neuronal cell membrane. Now, a chemical concentration gradient means that there is an unequal concentration of these different ions on the inside or the outside of a neuron relative to the inside or the outside. To maintain these chemical concentration gradients requires a lot of energy, and in particular, sodium and potassium exist across steep concentration gradients across the neuronal cell membrane, and those concentration gradients are maintained by a pump called the sodium-potassium exchanger, which pumps sodium and potassium inside and outside of the neuron in order to maintain the chemical concentration gradients. This is why the nervous system uses so much energy, even when we don't consider it being particularly active. All right, we're going to talk about then the concentration of different ions inside and outside the cell membrane of particular neurons. So we've got 5 different types of ion. Sodium, there's lots more on the outside than on the inside of a neuron. Potassium, the opposite. Calcium and chloride, there's more of those on the outside than there is on the inside. And then A- refers to big organic anions, things like nucleic acids and protein molecules, which have an overall net negative charge, and there's way more of them on the inside than there is on the outside from an electrical perspective. All of these different chemical concentration gradients then result in there being an electrical difference across the neuronal cell membrane. It's positively charged relative to the outside, I should say, is positively charged relative to the inside. We can measure the size of this electrical difference in millivolts, and at rest the size of that difference is 70 millivolts. You'll commonly see it represented as minus 70 millivolts. The minus doesn't really matter, that just tells you that the outside is positively charged relative to the inside. Were the inside to be positively charged relative to the outside, then it would simply be 70 millivolts, assuming that all the chemical concentrations were inverted. Basically, don't worry too much about the minus, worry about the overall size of the number. Minus 90 is a bigger electrical difference than minus 70, for example. What we're going to do then is we're going to talk about gradual changes in this cell membrane potential over the course of a few milliseconds. So we're going to start at minus 70, we're then going to go through 0 up to plus 40 and then back down again, overshooting and coming back to the resting membrane potential. This characteristic curve, which shows changes in the difference of the electrical charge across the cell membrane, is characteristic of an action potential, and we're going to work our way through all the different components of it. All right, those components then comprise the action potential. The neuron is ready to fire. The firing of an action potential starts at the dendrites, and in particular it starts with the binding of excitatory neurotransmitter to specific receptors located on the dendrites. To understand what happens, we need to take a closer look at the concentration of sodium and the particular ion channel through which it travels on the neuronal cell membrane at the dendrites. So here we've got extracellular and intracellular, and spanning across the neuronal cell membrane, we have these ligand-gated sodium channels. Sodium, as we discussed, is in much more higher concentration on the outside of the neuron relative to the inside. And ligand-gated refers to the fact that this channel will bind to a ligand, in this case, a neurotransmitter, an excitatory neurotransmitter. You can see the binding site for the excitatory neurotransmitter on the ligand-gated channel. Very simply then, all that happens is this excitatory neurotransmitter binds to the binding site. That causes a conformational change in the channel. It pops open, and once it's open, then sodium is able to flood down its chemical concentration gradient. Nature doesn't like these concentration gradients. It wants there to be the same concentration of sodium on the outside as on the inside. At rest, when all these channels are closed, sodium moves only very slowly across the lipid cell membrane. But when this ion channel is opened, there's a very specific hole through which sodium can travel very easily, and so it does so quite quickly. If we consider this then again in terms of our table, what we've got here then is sodium in high concentration on the outside traveling to the inside where it's in low concentration. Sodium is positively charged, and so from an electrical perspective, we have an ion that's positively charged moving from a space that is positively charged into an area that's negatively charged. Overall, then, this means that the positive charge on the outside starts to reduce, and obviously it cancels out then the negative charge on the inside. So the electrical difference across the cell membrane starts to change. It starts to come down as sodium starts to come into the neuron. We can see this then in the shape of our action potential. As some sodium starts to come in, then this minus 70 starts to reduce. If it gets to a particular threshold membrane potential of minus 55, then something different happens. Enough ligand-gated sodium channels open, enough sodium comes in, the resting membrane potential starts to come up towards minus 55. This triggers the opening of voltage-gated sodium channels. These voltage-gated sodium channels are located in high density at the axon hillock. Voltage-gated sodium channels work very much like ligand-gated sodium channels, except that they don't bind a ligand. Instead, they have this voltage sensing subunit which spans across the channel inside and outside the neuronal cell membrane, and it's able to detect this resting membrane potential. So here we have this voltage-gated sodium channel, and at minus 70, the voltage-gated sodium channel detects the membrane potential, and at minus 70, it stays closed. It can detect, though, when enough of the ligand-gated sodium channels have opened to cause the membrane potential to reach minus 55, and when it reaches minus 55, it pops open. Sodium then floods down its chemical concentration gradient. It's exactly the same as if it were ligand-gated, but it's been opened by this change in voltage, this reaching of the threshold. There are lots and lots and lots of these voltage-gated sodium channels on the axon hillock. So many, in fact, that once they all pop open at minus 55, then we enter this fast rising phase of the action potential. So much sodium comes in that we go shooting past zero, and we end up inverting the electrical charge across the cell membrane because so much of this positively charged sodium has entered the inside of the neuron. This plus 40 then represents the peak of the action potential. And at the peak, two things happen. The first then is that the voltage-gated sodium channels close. This means that no more sodium can come in down its reducing chemical concentration gradient. The second thing that happens at the peak of the voltage of the action potential is that voltage-gated potassium channels open. Now, the actual trigger for opening these voltage-gated potassium channels was the reaching of the threshold, just as it was for the sodium channels. But the potassium channels open on a 1 millisecond delay, so they open then at the peak of the action potential. What we then have now is potassium, which is, let me just show you, a positively charged ion, leaving a space that is now positively charged and going to the outside of the neuron along its chemical concentration gradient. This then starts to restore the original electrical charge across the neuronal cell membrane. We are in now what's called the falling phase of the action potential. So much potassium leaves the neuron that actually we overshoot again. We overshoot the resting phase and we end up down at around -90 or so. The sodium-potassium exchanger then gets to work to pump sodium and potassium back to their original places. This phase is called the refractory period. The refractory period is so called because even though the membrane potential, the electrical membrane potential, has got back to more or less where it was to begin with, sodium and potassium are in the wrong place to be able to fire an action potential. So during the refractory period, a neuron cannot fire an action potential. It needs to wait for the sodium and potassium exchanger to pump sodium and potassium back to where they were when we started. Okay, we're going to go back then to our neuron, and we've talked about the movement of sodium and potassium, for the falling and rising of the action potential, but we haven't really talked about how the action potential moves down the axon in this direction, as we've indicated on a previous slide. The means by which this happens is fairly straightforward. There are voltage-gated sodium channels and voltage-gated potassium channels located all along the axon. So here you have them, the first voltage-gated sodium channel and a second voltage-gated sodium channel. Very simply, all that happens is that the first voltage-gated sodium channel, the axon hillock, those all start to open once we get to threshold, and then the sodium floods in, and it goes into the neuronal cell membrane, across the neuronal cell membrane, and it then diffuses along the axon, and it diffuses along the axon, that causes a local change in the resting membrane potential. If enough of that happens and it reaches threshold, then the next voltage-gated sodium channel pops open, and the same process is then repeated all the way along the axon. The voltage-gated potassium channels, they're just the same thing is happening with them, but obviously the potassium is moving in a different direction, and they are moving along opening along a delay, and so the action potential is able to travel along the axon. Okay, we're going to talk briefly then about these myelinating cells. These are located on certain neurons along the axon. Myelin is very basically electrical insulation tape. The cells that make the myelin, there are different types of those depending upon the location within the nervous system. But in both cases, the myelin is largely composed of fat, 80% fat with a little bit of protein, and so it appears white. In our neuroanatomy videos and in neuroanatomy sections of your textbooks, you're going to be reading about and hearing about white matter. That is basically just neurons which have myelin. There are lots of regions of the brain where lots of the neurons are myelinated and they appear white. Regions of the brain which don't have many myelinated neurons, they would appear not white. Normally they would appear gray, and so we talk about white matter and gray matter. The main myelinating cells of the central nervous system, the CNS, are called oligodendrocytes. The main myelinating cells of the peripheral nervous system, the PNS, are called Schwann cells. All right, now we're going to talk about what happens when the action potential gets to the axon terminal. It will result in the release of neurotransmitter, and that neurotransmitter will then act on another neuron, the next neuron in the sequence. And the gap between those two neurons is called the synapse or the synapse. Ahead of the gap, then we have the presynaptic neuron, and after the gap, then we have the postsynaptic neuron, and the neurotransmitter travels in between the two. Neurotransmitter is a chemical, normally a small molecule chemical such as glutamate, GABA, or dopamine, or serotonin, all of which we're going to talk about in more detail in other videos. The switch then between this chemical signal of the neurotransmitter that travels between neurons and the electrical signal of the action potential that travels within a neuron, that switch is made possible by the channels which conduct the third ion in the five that we're going to talk about in this video, and that is calcium. In particular, voltage-gated calcium channels turn the electrical signal of an action potential into the chemical signal of a neurotransmitter. So here then we have our axon terminal. We have a presynaptic terminal and we have our postsynaptic terminal. We have these bags of neurotransmitter located in the presynaptic terminal, and you can see then on the outside of the neuron we have our voltage-gated calcium channel that actually spans the neuron. It has that same voltage sensing subunit as the voltage-gated sodium channel, and then we have these little purple stars, an abundance of calcium ions, way more on the outside than on the inside. So our action potential travels down, It pops open, then the voltage-gated calcium channel, calcium floods in and triggers the release of the neurotransmitter into the gap between the presynaptic and the postsynaptic neuron, known itself as the synapse. Now, that neurotransmitter, represented here by these little yellow dots, can do one of two things basically. If the neurotransmitter is an excitatory neurotransmitter like glutamate, then if enough of it is released and enough of it binds to the ligand-gated sodium channels on the postsynaptic terminal, then it will trigger the firing of an action potential by the postsynaptic neuron, i.e. exactly the same thing will happen. There's a second major class of neurotransmitters though, and those are the inhibitory neurotransmitters, things like GABA and glycine. They make action potentials less likely to be fired by the postsynaptic neuron. To explain how and why that is, we need to go back to the biology of our ligand-gated sodium channel. We talked about the ligand-gated sodium channel being located on the dendrites of a neuron, and when the neurotransmitter, if it's an excitatory neurotransmitter, binds, it causes the flooding in of sodium, and that can, if enough of it happens, trigger the firing of an action potential. If the neurotransmitter is an inhibitory neurotransmitter, then the same basic principle happens, except that the inhibitory neurotransmitters bind to channels that are ligand-gated by that inhibitory neurotransmitter, but those neurotransmitter channels conduct chloride. Now, chloride, again, there's a lot more of it on the outside than there is on the inside, and the same basic mechanism happens, that the inhibitory neurotransmitter binds to the channel, pops it open, and then chloride can flood down its chemical concentration gradient. However, chloride is negatively charged, unlike sodium, which is positively charged. So here you have chloride in high concentration outside the neuron, coming down its chemical concentration gradient into the neuron, moving from an area that has a net electrical charge that is positive into an area that has a net electrical charge that is negative. So negatively charged ion leaving a positively charged area makes that more positively charged, and a negatively charged ion moving into an area that already has a negative charge is going to make that even more positively charged. What happens then is the overall resting membrane potential moves from -70 down to about -85. That means that it becomes hyperpolarized. The actions of this mean that were an excitatory neurotransmitter to bind to this hyperpolarized neuron, much more sodium would have to come in order to trip that neuron up towards threshold. So our dendrites, they have receptors both for inhibitory and excitatory neurotransmitters. If an inhibitory neurotransmitter binds and chloride floods in, the neuron becomes hyperpolarized, which means that much more excitatory neurotransmitter would have to bind to those same dendrites to cause much more sodium to come flooding in to allow the neuron to reach threshold. All right, let's move on then to a summary of everything that we've talked about. Summary of an action potential: an excitatory neurotransmitter binds to its receptor. That's a ligand-gated sodium channel. When that opens, sodium comes in. There's a local depolarization. If enough of that happens, then it can trigger an action potential, which is the opening of voltage-gated sodium channels and potassium channels, although the potassium channels open on a delay. This then causes the action potential to travel down the neuron, and it causes the release of neurotransmitter. The neurotransmitter travels across the synaptic cleft and binds to the receptor, and if that neurotransmitter is an excitatory neurotransmitter, then the whole process is triggered again. If that neurotransmitter is an inhibitory neurotransmitter, then it hyperpolarizes the postsynaptic neuron, and that postsynaptic neuron is less likely to fire an action potential. Okay, that's it for this video. There's some credits for the images that we've used and edited in this video. Any questions, please do send me an email, p.newton@swansea.ac.uk, and I'll see you in another video. Bye-bye.